So, welcome everybody. This is Peter Bauer from CENTOGENE. I'm the Chief Scientific Officer CENTOGENE, and I'm happy that you dialed in, joined for this webinar dealing with genetics in Parkinson's disease, where I would like to share lessons from a large global cohort with you. So, you can see in the picture, CENTOGENE is a company based in Germany at the Baltic Sea in Rostock. You can see our headquarters here with the X on the roof, and that's where our production is located and where we receive all the samples, filter cards for genetic and biomarker analysis. Our mission and goals are to improve the lives of patients with rare hereditary diseases and, of course, to provide a precise medical diagnosis. So we are committed to support the patients and referring physicians lifelong and provide them best service in diagnostic quality, but as well support in taking the right therapeutic decisions. In general, it's our goal to transform global genetic data into medical solutions. And I think Parkinson's disease is really just at the edge, being a disease where we can show that genetic insights and as well new therapies will play together in a better future for these patients. Now, CENTOGENE is positioned as a rare disease company. More than 350 million people worldwide suffer from rare diseases, and most importantly, many of these diseases are hereditary, are early onset, and so especially children face severe disease with being even compromised in all their milestones, and up to 30% of these children will not even see their fifth birthday, which means the disease burden is really tremendous. So to change that for the families and the children is, of course, a big challenge and goal for us. In total, and that's surprising, more people die from rare diseases than from cancer. This tells us that we have to invest more focus, more money, more resources into bringing cure to these diseases, since the comparison with cancer, the whole area is still under-resourced. Now, I think the final intro slide is that to help with understanding rare diseases, CENTOGENE has worked for the last 10 years on building up a data repository, which is CENTOMD, the mutation database, meanwhile comprising more than 375,000 individuals, where we have seen genetic variants, phenotypes, and as well biomarkers correlating for insights into the pathophysiology and, of course, the epidemiology of these diseases. So we can derive from there faster diagnosis and a better clinical picture than you can get from textbooks. Now, the topic today is Parkinson's disease, and Parkinson is a very common late onset neurodegenerative disease where patients face three major movement disturbances. One is bradykinesia, which means it's hard to initiate movements, and so for those patients to change something from being in rest and start activation is hard to initiate. Furthermore, they have resting tremor, especially at the upper limbs, which impairs their motor skills, writing, eating, doing manipulations, and furthermore, the tonus of the muscles is increased, which is called rigidity, and this rigidity leads to a special posture, cantochormia, but as well the rigidity impairs walking and provokes frequent falls. So overall, this is a progressive disease, and patients start to deteriorate over years. There's good established treatment which interacts with the dopamine circuits in the brain, and pathophysiology is that cells in the substantia nigra degenerate, and the whole regulatory circuits to have these fine movements is degenerating over time. From an epidemiological perspective, we know from a population-based study published more than 10 years ago that Parkinson's has a prevalence of more than 5% in the general population, and we can see that this prevalence is clearly age-related, so having roughly 1 to 2% in the 70-year-olds and rising up to 10% in the 90-year-olds. So this is a disease burden of the aging societies, and so to understand and treat symptoms of Parkinson is very important for kind of the general health of aging societies. If we look into etiology, there is 3 major contributors. One I already mentioned, which is just age. So the older the population gets, the more frequent we have symptoms and signs of Parkinson's. Then we know exactly that there are neurotoxins, for example, pesticides or some ions like manganese that trigger the degeneration of neurons, and then the patients start having PT symptoms. So in general, that's a destruction of the dopaminergic neurons, which can be due to toxins. And then there's many genetic factors, which I will review in more detail with you. The main genes being LRRK2, the leucine-rich repeat kinase 2, synuclein, alpha-synuclein, which was the first monogenetic cause of Parkinson's described in the 90s, and PARK2, leading to Parkin dysfunction, which is the most common autosomal recessive form of Parkinson's. There's other more rare forms of the disease, and you can see here, if we look in more detail into the monogenetic etiology of Parkinson's disease, we have basically 3 genes that cause classical Parkinson's disease, which means hardly to discriminate between the idiopathic non-genetic form and Parkinson's caused by synuclein, LRRK2 or VPS35 mutations. All 3 are dominant, while synuclein is highly penetrant. We see reduced penetrance in LRRK2 carriers, for example. Then we have a rarer group of early onset Parkinson's disease, so manifesting before, usually before the age of 50. PARK2 mutation carriers, which have an autosomal recessive disease with a B-allelic compromise of the protein, are the most common ones, and rarer autosomal recessive forms include mutations in PINK1 or DJ1. And then we have a couple of atypical Parkinsonism syndromes where the clinical picture includes as well other motor system or non-motor systems like ATP13A2. We have a very prominent dystonic contribution, and others where cognitive decline and other neurological systems are involved. Now, a very particular story in Parkinson's is the contribution of glucocerebrosidase gene variants. GBA is the name for the gene. And what we learned already in the late 90s is that families that usually have been identified because homozygous mutations in GBA cause Gaucher disease have a higher incidence of Parkinson syndrome patients on top, which means in this pedigree, there is 2 individuals, a female and a male with Gaucher disease, but there is a couple of at-risk patients that are heterozygous carriers of the GBA variant that show signs of Parkinson. The frequency of Parkinson is as well increased in homozygous Gaucher patients, but over time we have realized that GBA mutations are the most important genetic risk factor to develop Parkinsonism. And other than recessive or mutations like in Parkin, the penetrance is not 100%, and so the demonstration of a mutant allele in GBA does not yet diagnose a disease. It's a risk factor like outlined here. And so if we look into a general concept how effect size and allele frequency contribute to a phenotype, we can clearly map Parkinson genetics into 3 major dimensions. One is the blue one here, where we have high penetrance for very rare alleles, which would include the recessive Parkinson syndromes. We have rather still low frequency variants, pathogenic variants in the GBA gene that have an intermediate effect, which means only 5 to 10% of the mutation carriers will develop Parkinson's. And we have complex risks, which are usually identified by genome-wide association studies, where the individual risk contribution, so the effect size, is only 10% or 15% over the normal population, which means those variants also identified don't have a high predictive value in an individual case. However, GBA is in between and still contributes a relative risk that is doubled in variant cases to variant carriers to increased 5 times over the normal population. If you assume a 70 years old has 1 to 2% risk of developing Parkinson's, if he has on top a GBA variant, you end up with 10, maybe 15% disease risk, which is significant. What did we learn clinically about these GBA patients with symptoms of Parkinson's disease? We learned that they are on average earlier with their onset. So while the wild type Parkinson's start to develop symptoms with 60 to 70 on average, GBA PARC patients have a shift towards younger age of 5 up to 10 years. And we've seen that this type of Parkinson seems a little bit more aggressive, which includes as well cognitive decline and marked disturbances of visual constructive competencies and working memory. So this seems to be a rather specific subtype. And since we know about the genetic etiology, of course, there's hope and the expectation that treatment as well can focus on the dysfunction that is triggered by the GBA heterozygosity. Now, there is similar or different, but similar findings for LRRK2 carriers as well. Here I can show you that the penetrance of the most common mutation in LAG2, which is the substitution of glycine to serine at the position 2019, has a huge variability in when and how many variant carriers develop symptoms. So we can see that in North Africa and Tunisia, where this variant is pretty frequent, the carriers tend to have early symptoms, and you can see that up to 50% develop Parkinson's by the age of 16, while the very same variant in the Norwegian background, so in Scandinavia, accounts only for 10% clinical symptomatic cases, while with 60, still 9 out of 10 Norwegian are without clinical signs. Since this is the very same variant, this indicates that modifiers of this manifestation might be found, and they can be genetic or environmental. Lot needs to be defined in these cohorts to know better how to predict individual disease risk for these patients or variant carriers in the future. So to summarize a global perspective on genetics of Parkinson's disease, I think these two genes, LAG2 and GBA, are by far the most important in terms of number of patients showing these variants and having symptoms of Parkinson's. For LAG2, a large gene, we have identified at least 9 evidenced variants that cause the disease with reduced penetrance, and we have evidence from cellular models and from in vitro analysis that these variants lead to an overactivation of the kinase. And so this is a gain-of-function effect that triggers the clinical picture. As said, we have a high risk and a high penetrance in North Africa, while the opposite is true in Scandinavia. There is a couple of cellular markers that are changed, phosphorylation of proteins, and so therapeutic targets have been looked after in these pathways, and a couple of new small molecules are around that might be tested in clinical studies or already tested in clinical studies that interfere with this pathological activation. For GBA, even more prominent, we still don't have an approved pathophysiological model and can't tell why heterozygosity for these variants lead to Parkinson's. However, since they are more frequent, we have more variants and more evidence on the genetic side. Interestingly, even variants that are not associated with the recessive disease, with Gaucher disease, lead to mild Parkinson's symptoms. And so it seems to be different from the pathophysiology we know in Gaucher disease. The risk, if a carriership for GBA is demonstrated, is 2 times to 5 times increased, and the phenotype is overall indistinguishable from idiopathic Parkinson's disease, with a predominance of cognitive symptoms in the later course, which might make it a little bit special after a couple of years. And as said, the onset is earlier than in the classical idiopathic forms of Parkinson's. If we ask the question how genetics should be added to the diagnosis of PD patients, of course, the earlier the symptoms, the more probable we will find a genetic etiology. And I can just share with you data from a recent study where we had a collaboration with the group at the University Hospital in Lübeck, Trin and Klein and colleagues. and did exome sequencing on patients with early onset Parkinson's disease and identified a couple of recessive early onset patients, like this one Parkinson patient, and as well some very rare differential diagnosis where Parkinson's symptoms have to be seen in the context of another neurodegenerative disease like NBIA. But not surprisingly, there were as well a couple of carriers of GBA alleles at codon 502, 483, and 252 that showed symptoms of Parkinson's, and so just told us as well in this study that GBA related Parkinsonism is the most common form. There's different assays to analyze genetic variants in PD patients. The most cost efficient would be a gene panel only focusing on the established genes of Parkinson. However, we learned from this study that usually the gene list that is applied is too small to capture all the variability in patients with early onset Parkinson's, and one conclusion is that exome sequencing in these patients was very efficient. Of course, when costs are going down as well, whole genome sequencing will be discussed because we wouldn't miss non-coding variants and a context where as well the regulation of genes will have an impact on the manifestation of late onset neurodegeneration. So at the moment, gene panel sequencing is a very good standard, and we've seen that in a couple of patients where the onset is especially early or repeated cases in the family have been seen, exome or genome sequencing should be sought after. Now, what did we deduct from this analysis, which was done in the last 2 years? We've seen that the regional variant in the disease penetrance is not understood. And on top, we do not understand genetic factors that modify penetrance for LARC2, for example. The same is true for GBA variant carriers. However, for the latter, we don't have even data that regional differences would exist. And of course, since we cannot predict whether a carrier is developing symptoms or will stay healthy, at least the first 80, 90 years, we need to develop better markers to predict the future phenotype, not from genetics, but from those markers, because the genotype itself did not instruct us in the past. So this is a call for diagnostic biomarkers, be it in imaging or in a laboratory analysis. And this was exactly the motivation why CENTOGENE started to organize a global study on Parkinson's disease called ROPAD. This is an international epidemiological observational study where we would like to understand regional differences, the genetic background, and develop biomarkers. A total of 10,000 participants are anticipated. You can have more details about the study and the protocol at the web pages given here. And we have pretty straightforward inclusion criteria, which is, of course, informed consent, participants clinically diagnosed with Parkinson's disease, or participants that are family members of a Parkinson's patient that is already diagnosed genetically, in this case LARC2 positive, or it's a participant from a high-risk population, which means Ashkenazi Jewish or North African origin, where these genetic mutations are highly enriched. Of course, participant needs to be older than 18 years, but in fact, since Parkinson's rarely developed before the age of 40, they are much older on average. And I will show you that. Exclusion criteria I don't mention, they are just stating the negative of the positive inclusion criteria. Now, what are the objectives? So by screening 10,000 patients with Parkinson's, we aim to identify 1,500 LAG2 positive patients. And of course, we want to study as well those that are non-LAG2 PD patients to have a control group. And the tertiary objective is that in the LAG2 positive cohort, we want to go for biomarker screening and establishment. The study opened in June 2019 and aims to include patients over 24 months. So we are just now in the first quarter of the total time of the study. And as said, I will share a couple of preliminary results with you. What do we do? So if the patient is recruited, which means the clinical picture has been captured and put into the ECRF, we will do blood sampling, and this will be this dried blood spot filter card you can see here in the chart. And from this sample, we punch a small piece of paper, do the DNA preparation, and sequence hotspots of LAG2. So that's established and discussed variants that cause Parkinson's disease. And we sequence as well the whole GBA gene to have a primary screen on those that have the most common forms of genetic Parkinsonism. If this test is negative, all patients will proceed, or our samples will proceed into a PD-associated gene panel, where we have assembled 47 diagnostic genes that are associated with Parkinson's, and in these genes we report pathogenic and likely pathogenic variants to the referring physician, so that after this second step, an account for all the established Parkinson disease genes is available. There is an option to move further on into whole genome sequencing for a limited number of patients, especially those where we have more than one affected in the family, or extremely early onset, which then directly points to a higher genetic contribution in this patient. Now, that's just one of the observations as an example that I can share with you how reporting looked like. So, of course, you get the patient information, which is coded, so it's not names, it's just a study code. But then within the study, in this case, we would report back heterozygous pathogenic variant in VPS35. You remember this was one of the dominant Parkinson disease genes that lead to a typical Parkinson syndrome. You get details of this variant, and especially we show you that there is evidence, and this variant has already classified in the literature to be pathogenic. So this is then given in the variant interpretation, where we say this is a heterozygous carrier, and we have since 2009 evidence and added in 2012 that this variant is in fact causing Parkinson's disease. You get some analysis statistics to share the quality of the analysis with you, and with that you can then talk with the patient on further decisions or learnings in the family. As said, this is the primary goal, to have LAG2 and non-LAG2 PD patients characterized. But the tertiary objective, I think, is even more interesting, where we take as well a punch from the filter card that is sent in with patient material with whole blood, and we run MRMS mass spectrometry in different groups, which is those that are LAG2 positives, those that are PD patients with other PD-associated mutations, and of course patients without mutations, and healthy individuals. And what we would like to do in this high-resolution mass spectrometric approach is to identify markers that are specific for any of these groups. This is a very fast and insightful analysis where we need just 3 additional punches from the filter card. And just to show you here from a preliminary screen in LAG2 carriers with Parkinson's, which was 46 individuals, we have selected 14 patients. And in the initial discovery phase, you can see it's different variants. We've pathogenic variants in LAG2 we have sampled. We have already 22 biomarker candidates upregulated and 8 downregulated, and are just now to validate these findings in additional patients. This technology is extremely powerful because it's with a high sensitivity for small molecules in the plasma of the patients, which is the filter card punches. And we see with the stratification for genetic background, of course, we have pretty small noise bands for controls and good dynamics for the patient values in these comparisons. Now, I would like to share as well a couple of first results from the ROPAD study. Meanwhile, I told you we have now the study open since July. More than 658 cases have been analyzed, 2/3 or roughly 2/3 male, 1/3 female. The mean age of the patients is above 65, and we have a large spread of onset or age of these patients from pretty young to very old. We have different study sites globally, so especially in Europe now, the first ones, and then as well in North and South America. And you can see that there's already a global footprint for the samples we have analyzed. And if we look into the first result for the primary goal, which is LAG2 carriers, we have seen 19 out of the 650 individuals with LAG2 mutations, which is close to 3%. You can see that they are rather older in age when they are diagnosed. The earliest or the youngest was 53, and most of them beyond 70, a couple even beyond 80. And for a subset of the samples, we have done the analysis of the gene panel, so those PD-associated genes, where we report on 47 genes, and in this cohort, we have seen a couple of recessive variants in PINK1 and in Parkin being the most prominent, and we have variants in POLK on the 3rd place. So the concept of doing this stepwise analysis has proven to be efficient, and beyond the classical Parkinson genes, we see as well syndromes that involve only Parkinson's symptoms. Now, what did we learn so far? Summing up all genetic findings, we have a genetic cause, or at least contribution in 15% of the cases we have looked at in these first months. The penetrance cannot predict for healthy LAG2 or GBA carriers, so we don't know how to do that without a biomarker, but as I have shown you, the biomarker program was very successful at the beginning, and we have already 30 candidates to further validate to come back with a meaningful predictive validated marker for the second phase of the study. Overall, this will be the largest epidemiological global study, and this is closely linked to a further study that is now set up at the University Hospital in Lübeck, Christine Klein's working group called ROPAD, and patients that have received genetic testing in ROPAD can easily be transferred to this scientific-driven study environment where questions about toxins and penetrance and of course in more detail clinical phenotyping of the patients will be done. So we think that this resource is very powerful and important to close knowledge gaps and of course as well identify and line up patients for future mutation-specific therapeutic trials. Having said that, I'm at the end of the presentation. Thank you for your attention, and still would motivate you first of all to use the Q&A tool of the software here to ask questions and on top answer the questionnaire that we will send out after the webinar so that we learn what you liked most or did miss in the presentation and give us an opportunity to improve as well the quality, the interaction with our webinars. So thank you for your attention, and now we can start looking into questions and answers if you would like to drop some. So I have a first question where the question is raised where the study centers are open at the moment for ROPAD, and I told you that we have a couple of centers in Europe, in Germany, in England, in the US, in South America, and you can just contact our clinical study teams using ROPAD at CENTOGENE.com and we'll get a reply where patients can be recruited, or if you're interested to collaborate and even become a study center, drop us an email there as well. Okay, there is another question on LAG2 genetic screening, whether the first test, the hotspot test, includes all the coding sequence of LAG2, which is a large gene, or is limited to really the mutations we see. The answer is we amplify those regions and so we see especially those described mutations in the first hotspot test, but of course see as well surrounding sequences. However, in reporting, we only will mention variants that are depicted in the list, which means a new variant seen in a patient is usually a variant of uncertain significance and will not be reported. This does not mean that in the future, if we have enough evidence to classify such a variant as likely pathogenic or pathogenic, we will not go back and as well report on a new finding, because this large study definitely will help us to classify rare variants more decently. There's a question from South Korea, whether we have a study center in South Korea, and I will just defer this as well to our clinical study team that will be in touch with you asking this question and talking you through possibilities to join the ROPAD study. Okay, there's another question about treatment, since I mentioned as well that molecular treatment is developed. Yeah, there's not yet, of course, an FDA-approved treatment, but we know that there is development. For example, Denali is working on a LRRK2 inhibitor that shows in early phases of clinical testing a good profile and is promising a promising candidate to achieve mutation-specific treatment in the future. Whether or not this will also help patients that don't have LRRK2 mutations, of course, is not at all predictable. And so we have to see how the clinical study program will progress and how efficacy, but as well potential adverse effects are handled for this type of drug. For GBA, there is currently a study open for substrate reduction therapy, which has been developed for Gaucher patients initially. There's a debate on the mechanism of action, and we have to see whether the rationale and then, of course, the clinical effect of this intervention proves to be efficient in Parkinson patients. Since we don't have clinical data on that, I don't dare to comment. However, I think there is still room for other mechanisms and new therapies dealing with the GBA pathophysiology. Okay, I think there is no other questions from the audience. So once again, kindly give us feedback with the questionnaire, or there comes in a last question I will answer. So LRRK2 gene mutation is with low penetrance is a question why it was preferred to shortlist patients for your study. Then MAPT. So if I understand the question correctly, the question is whether the penetrance of MAPT patients, which is high, would not give a clearer genotype-phenotype correlation. Yes, that would be the case. However, even though LRRK2 penetrance is low, the frequency of LRRK2 carriers is high, and so in total, we see more LRRK2 carriers with Parkinson's symptoms than MAPT carriers with Parkinson's symptoms. The story might be different for the complex multigenic contribution of microtubulin-associated protein tau, since we know from GWAS studies that this locus plays a role as well in idiopathic Parkinson. However, at the moment, there is no clear concept how this can be tested functionally, and therefore we just don't have access to this question in the patients at the moment. Might be that this will come up, and then still, of course, some predictive variants can be tested in the cohort, and the correlation can be done even later if there is no evidence. Okay, so thank you all for your interest in our story on global insights in Parkinson's disease. I once again remind you to give us feedback on the webinar, and we will have additional webinars in the new year dealing, especially focusing on biomarkers. And so I would be glad if you would join in then again. So thank you for today, and all the best. Bye-bye.